Okay, so good morning, everybody. Welcome to the 28th Structure Meets Function webinar, which this month is going to be hosted from Instruct Finland. We've got a couple of great speakers who are going to be talking to you today who have accessed some of our facilities in Finland. So for those of you who don't know, I'm just going to do a brief introduction to Instruct-ERIC, which is who we are. So we are the structural biology research infrastructure, which means that people can access advanced high-end structural biology services from any of our centres across Europe. We also have a number of research sites for training purposes, and then through Instruct as well, there are several other funding opportunities such as internships for early career researchers and also R&D funding. In terms of funded access to Instruct, something that may have become apparent recently is that the funded access call is closed for 2023, and this is going to reopen in early 2024. But there are several other opportunities to get funded access to structural biology services. One of those, for example, is CanServe, which has very recently opened its open call. This provides free access to all Instruct structural biology services for cancer research. So if you have any cancer-related research projects, do get in touch with CanServe, do check out the website and have a look at the open call, and you can find any of the services there. There are also other projects such as Isidore and iNext Discovery, which again provide access to Instruct services through those as well, funded access. So just a bit of housekeeping there, and I'll pass over to Jan now, who'll introduce our speakers.
Okay. Thank you, John, and warm welcome from my side as well. So I am Jan Enis from the Department of Chemistry at the University of Eastern Finland, and I will be chairing this webinar today. And today we have two speakers, Dr. Ilona Rissanen and Giovanna Viola. So welcome both. And so Ilona will be first to start. So she's an academic research fellow at the Institute of where her group studies structures and mechanisms underlying viral infections and immunity. And today she's going to talk about some novel small protein inhibitors, potentially neutralizing SARS-CoV-2 virus, which is well known to all of us. And also in this work, the understanding of how these inhibitors target the spike protein was achieved through cryo-EM. So means that some of the Instruct-ERIC facilities played a pivotal role also in this study. And so, floor is yours.
Well, thank you, Janne. Thank you, organizers, and thank you, everybody, for your attention. I'll be very excited to talk about our recent project. I will start sharing my screen, which you should see my presentation now. Good. So going forward, and this will be a talk on a small protein inhibitor for SARS-CoV-2. My group works on viruses, particularly their envelope architectures and entry structures. And here you can see some of the beautiful world of viral structures. I hope this inspires you to consider these as attractive targets going forward. Presented here, HIV, Ebola, and flaviviruses, though today we will be talking about coronaviruses. So as Janne mentioned, they don't really need an introduction, but I'm going to give you a little bit of it regardless. So about the molecular architecture of SARS-CoV-2, the causative agent of COVID-19, it is a positive sense RNA virus with the genome enclosed in a lipid bilayer envelope, where you then have trimeric spike proteins as the outermost viral component. And these are highly relevant as they mediate post-cell attachment via the ACE2 receptor and other receptors as well. And then also they mediate fusion once viruses have been taken into the endosomes, and they can also mediate fusion at the cell surface directly in the right pH conditions. So this protein is the main component in for therapeutic development and also vaccine development. We've all received RNA vaccinations, most likely from certain producers, encoding the S protein, as that is the main target of neutralizing immune response. So here we'll go a little bit into how this protein functions and how those functions can be prevented. And I hope to bring some new insights. I think there is a lot of work that has been done on this topic, but there are still certain features that are not as well understood. And this is what we will be digging into today. Our starting point is that we had an excellent collaboration here in the University of Helsinki, where the group of Professor Kalle Saksela, where Anna Mäkelä was heavily involved as well, developed this novel type of small protein inhibitor. And this is a very tiny protein, smaller even than nanobodies, only 6 kilodaltons in size approximately, and it's based on the SH3 domain from human nephrocysteine, where they have made a phage display library where 2 loops of this domain are variable sequences, and then you are probing different targets in there and fishing out inhibitors that bind to them. They started doing this when SARS-CoV started outbreaking, and they were able to fish out certain inhibitors that seem to have binding properties to a lot of different RBDs, also from the variants of concern, despite these having accumulated a lot of mutations by this point, lots of amino acid changes. So these went forward to characterization with various collaborators here in the University of Helsinki and also at University of Zurich. And what was found was that this SH3 domain-based inhibitor, when fused into a 3-module assembly, so there is an SH3 domain, and then a linker, another SH3 domain, and linker, third SH3 domain. This is to take advantage of the trimeric nature of the spike glycoprotein, so we get the avidity benefit of having trimodular binding, targeting a trimodular spike protein. This construct called TriSP92 had very high IC50, so half maximal inhibitory concentration against all extant SARS-CoV-2 variants and even the original SARS virus from the 2000s. Quite interesting. So here you can see the IC50s, which are sub-nanomolar in most cases, and even low nanomolar for the original SARS virus. And as this study was done mostly during the last year, we had all the variants up to the XBB in there. So not the very latest Omicrons, but a good array of all the Omicrons there. And as you can see, IC50s remain excellent. And for structural biologists, then I entered into this project with the question of how is this? Where does this inhibitor bind? What is the site that can be targeted to get such broad, potent neutralization of these coronaviruses? And here I want to mention our paper that came out earlier this year in March in Nature Communications. So most of the work that I'll be talking about today is of this work. So if you are interested, please take a look. It is available. And before we go into the analysis in more detail, I wanted to mention the infrastructures that really make our research possible. So as structural biologists, we really need top-level infrastructure available for our research. And here locally, we are users of the cryo-EM unit. We also do a lot of protein crystallization. We use the facilities at the electron microscopy unit and the biocomplex purification as well. For data processing, CSC in Finland is a really excellent platform, and internationally, Instruct-ERIC is really invaluable, as is also access to synchrotrons. And then we also use really nice microscope facilities available at SciLab lab in Sweden. What I particularly wanted to mention is that the cryo-EM unit here in Helsinki, headed by Sarah Butcher, was crucial for our sample preparation, because I started producing a viral spike glycoprotein for coronaviruses in early 2020. And even though at that point there was a bit of insight into what to put in your design in order to make it as stable as possible, it still took a good amount of time to get sample prep working. And we were able to screen our grids locally, just see what's working, get the optimal preparations, optimal concentrations for high resolution data collection. So without that option, I think work on unstable glycoproteins like these would really not be possible. And the cryo-EM unit is part of the Instruct-ERIC network. So really big thanks to that platform. So going forward, I will not be talking about the sample preparation itself in detail, because a lot of work is already out there on how you might produce spike proteins. The important thing to keep in mind, if that's something you are considering, is that for structural biology, you want your proteins to be in the native conformations. And as full glycoprotein spikes, they mediate fusion processes, so they are conformationally metastable. And what we noticed in particular was that if we store this protein, even for a short while, we see much fewer particles in the pre-fusion conformation and significantly more particles that have just lost their ordered structure. So if you are thinking of producing this protein, consider producing it via transient expression. You collect your supernatant, purify it, and then immediately put it on grids. We spend a lot of time figuring that out. Also, which tags you're using is quite important. With histidine tags and nickel affinity purification, we got a bit more aggregation. Then later on, we moved on to using strep tag, and that worked a bit better when it came to getting particles that were individual. For data processing, so once that optimization had been done for being able to get SRAM or preps that were of good enough quality for structural resolving, we started data collection. This was done on a Titan Cryos microscope in Sweden from the SciLife lab, accessed specifically in Umeå. So many thanks also to Michael Hall, who was helping with this data collection. We did an overnight data collection there and got a nice number of micrographs. We did see some aggregation of particles. These were preps still produced with his tags and purified by nickel affinity, and there this behavior is common, but there was a good number of particles that could be picked regardless. So in order to get the most out of the data, we did first just block picking. After classification, that gave us an idea of the particles that we could then put forward to TOBAS training. So just the best micrographs where most particles were picked, we used that in training, did a couple of rounds, and we're able to then get the TOBAS algorithm, which is a kind of a learning algorithm for particle picking, where we're able to get a good number of particles from that data. So that really helped. I recommend using that if you are running your data processing in CryoSpark, as we did. And going forward, the next step that might be of particular interest for people working with SARS-CoV-2 is the variability analysis. So I'm just highlighting that as an option for distinguishing between your RBD conformations. As in SARS-CoV-2, you're going to have up and down conformations in different proportions, and in different RBDs being up, different RBDs being down. You can have 1 up, 2 up, or 3 up, or all down. And in order to isolate these populations from each other, you will need to use some 3D classification or variability analysis in order to get as good a resolution as you can in the RBD. That can still be quite difficult using cryo-EM to get a high resolution for the RBD due to its flexibility, but we were able to significantly improve our outcomes by using the variability analysis, where we then got populations that corresponded to a mixed population of up and down RBD states, which was the one with the least good resolution. So our analysis then, we focused on a significant number of all up RBD conformation particles that we saw, and also the minority population, which was all down particles, and reconstructed these, ending up at a resolution of approximately 3 for both full spike reconstructions. However, this was mostly in the S2 core unit. The most ordered part of the particle was, of course, the one to have the highest resolution, while the RBDs that were of our interest, since that was what the inhibitor was produced with, so the phage display library was panned with the RBD. We knew that that is where the inhibitor is binding, but those regions being quite flexible, we then needed to do a local refinement, masking initially out other parts of the particle and really just focusing our refinement on the RBD region, which at least helped us to get consistent features at approximately 7 angstroms resolution, where then fitting becomes feasible. Going forward, just to show you what sort of density features we observed, was we got the all-up RBD conformation with very clear extra density here colored orange that does not correspond to any part natively in the S-trimer itself. So we immediately we thought, okay, this is our inhibitor, but I will go into showing that in a little bit more detail later. The fact that we saw so many all-up particles, so majority population of S-trimer in the all-up conformation is highly unusual for the SARS-CoV spike. Usually it is either all down or one RBD up and two down. These are the most common states seen, and all up is extremely rare unless bound to, for example, the ACE2 receptor or to an antibody that enforces or facilitates an all-up conformation. There was an all-down conformation where we did not observe any extra density on the RBDs, and then there was a mixed up and down conformation where we observed extra density adjacent to the one up conformation that was observed. And upon getting a higher definition, because from the previous slide, I hope you can tell that the RBD and servo body density, the inhibitor density, is a little bit blobby, a little bit fragmented. But upon our local refinement of this, we were able to get to a resolution where the RBD could be fitted with very high confidence. And to our great delight, also the SH3 module was something that could be fitted in here with high confidence. So what we did was there is a crystal structure or an NMR structure of an SH3 module that has been reported, and that was used as a basis for making a prediction of the servo body with the proper sequence in there. I should note that AlphaFold did not do a good job here, so we also attempted to model the inhibitor with AlphaFold, but because that inhibitor has highly divergent sequences in these two loops that have been completely modified, then AlphaFold was not able to predict any sort of SH3 type fold from that. So we used the existing models and also iTASR to produce a prediction of our inhibitor using the well-known architecture of SH3 domains. And here you can see the fitted RBD matching the density features. There is even extra density where there is a glycan at the asparagine 343. So our placement very nicely matches the features that are ordered in the RBDs. The very loop of the RBD at the top there, that is flexible, so we're not surprised not to see density for it in the up conformation. When it comes to the servo body module, we performed this fitting in Chimera with global fitting. We did not do any manual placements, just global fit map search, first placing the RBD, then splitting the density so as to keep the RBD density reserved for RBD, then placed the servo body inhibitor in there as well. And what we got was a very nice fit, particularly of the RT loop, which is one of the modified loops in this construct that was placed at the interface with the RBD, and that was very clearly the most ordered part of this inhibitor bound. The other modified loop we didn't see good density for in the cryo-EM reconstruction, so what we think is happening here is this specific inhibitor primarily binds via the interactions mediated by the RT loop. Of course, the resolution is still quite limited, so while from this we can tell where the inhibitor is binding and its binding footprint, its epitope to an extent, I would not go so far as to start talking about the side chain interactions that are mediating this. But we can definitely place it and we can see where it is in the context of the spike. So here, going forward with the analysis, what we can tell is that it is binding at the site that then enforces an all-up conformation because this site is incompatible with being in the down conformation as long as the servo body is bound. Normally, the site where the servo body binds in the down conformation is closely packed with the other RBDs and with the helical core structures of the spike. So there would be no way to sterically allow both servo-body binding and down conformation at the same time. Our running theory is that as the RBD sample up conformation, then servo-body will bind, and then it will prevent reversion to down state. And as soon as another RBD samples an up state, remembering that the inhibitor is a trimer, then one of the free modules is going to bind there. Then it keeps two RBDs in the up conformation, and once the final one again samples up state, it is going to be sequestered in the up state. And this is supported by the reconstructions that we got from the data, having a small minority population of an all down, a large majority or significant majority of all up, and then in the mixed population, the single up conformation that was present did have the servo body module bound. So I would think that then represents an instance where it's just waiting for the other RBDs to sample the up state in order to be able to bind and sequester the full all-up conformation. Here we are also showing that the inhibitor does not compete with ACE2 binding based on the structural analysis. So the ACE2 binding site, the receptor binding site, is distinct from the footprint of the inhibitor. And importantly, we also see that the mutations that have been kind of concentrating into the latest variants, amino acid changes that have been occurring up to the XBB variant are elsewhere than at the SB servo body binding site. We believe that this is because a lot of this is accrued at the immunodominant sites where antibodies in the classes 1 and 2 of the RBD epitope classification bind, largely overlapping with the ACE2 binding site. While in our case, the epitope seems to be quite conserved and only really peripheral residues are seeing amino acid changes. This is likely to underlie the high cross-neutralization potency observed for this inhibitor. If we look at it in a little bit more detail, we can here see in the sequence alignment, we have SARS-CoV-2 and SARS-CoV-1 sequences. And the color blocks denote the binding site of our inhibitor in orange, and then the binding site of the ACE2 receptor in purple. And a lot of the mutations are occurring within or proximal to the ACE2 binding site, but then our inhibitor binding site is mostly remaining free of mutations. And importantly, between SARS-CoV-1 and SARS-CoV-2, there is only a single amino acids change when it comes to the epitope of the inhibitor. So we believe this also underlies the capability of the inhibitor to inhibit original SARS virus. I want to highlight here the work of my PhD student Liina Hannula, who was involved in all of this work here. So the data processing, the analysis, mapping all the escape mutations, and so on, and then also in figuring out the potential neutralization mechanism of this inhibitor, which is presented here. So importantly, now from structure, we know that it's likely to inhibit by a mechanism that is independent of ACE2 binding. And this was confirmed by Kalle and his group as well in functional studies. So they tested for the capacity to of the inhibitor-treated viruses to bind ACE2 displaying cells, and this capacity was retained even after inhibitor treatment. So we know that the viruses treated with the inhibitor can still bind ACE2, but they cannot undergo further steps in the cycle leading to infection. So we believe that this enforced all-up conformation results in a fusion-incompetent spike conformation. This is a theory that has been put forward also for antibodies that bind this region. So one of the best-known binders of the so-called cryptic epitope is the monoclonal antibody CR3022, which is an antibody for SARS, the original SARS virus. And intriguingly, so this antibody also binds the SARS-CoV-2 RBD, but it does not really neutralize the virus particularly well. It can neutralize SARS virus, it binds this conserved region, but it does not particularly well neutralize SARS-CoV-2. And that was I think the reason why originally this was considered cryptic epitope. It wasn't really known what binding to this does, and also where these differences come from. Maybe now we can shed a little bit of light on it. So we know for a fact that it's not a thing of ACE2 binding that the inhibitor is preventing. Instead, it causes a conformational change. In our cryo-EM data, we see the beginnings of this conformational change in the enforced all-up conformation. I did not include it here, but in a very large data set, we also see some particles starting to open drastically. At that point, of course, you lose a lot of resolution, but it does look like even in these prefusion stabilized constructs that are being used for structural analysis, the spike is really starting to lose its functional prefusion conformation after treatment with the inhibitor. Then the question of, okay, why is this inhibitor so potent when antibodies that target similar conserved regions really have significantly lower IC50 values, and even if they have good RBD binding, may be completely unable to neutralize SARS-CoV-2. And the reason here, I think, is related to the spike opening dynamic. So as mentioned, there is that tendency of the spike to sample these RBD up and down states, and even to open further. And here I want to highlight a beautiful molecular dynamics study by Zimmerman et al., where they did a very long, I think 0.1 second long molecular dynamic simulation using huge computational resources. And they were able to find that the original SARS spike opens to a greater extent, if you allow it time in these simulations, it opens to a greater extent than the SARS-CoV-2 spike. And this would potentially explain why this antibody CR3022, which targets a conserved site, but a site that is inaccessible in the down conformation, can be targeted in SARS-1, but not in SARS-2, as SARS-2 is more closed overall than SARS-1. In our case, because we are not working with an antibody, we are working with a particularly small inhibitor. If we consider antibody a Fab fragment to be 50 kilodaltons, a nanobody 12 to 15 kilodaltons, and this is only 6 kilodaltons with a small binding interface. We believe this is the reason why our inhibitor can target the epitope. It's small enough that even a spike that is largely closed can allow the binding of this inhibitor. So as a few key takeaways, we have the cryptic epitope on the RBD that is highly conserved, but it is sterically quite challenging to reach for conventional antibodies. They can reach it, and there is some evidence of them then starting to really force the spike open, so really destabilizing it. But these antibodies tend to still have lower potencies. While in our case, as we developed a small protein binder that is also extremely stable, so these SH3-based inhibitors, you can keep them at room temperature for a year just in a tube, and they are still going to retain full potency, so significant potential there. And also they are resistant to aerosolization, so you could take them potentially via an inhaler directly to your respiratory pathways to prevent infection of respiratory pathogens. As they are so small, they can access the site and potently inhibit. And we want to highlight the fact that these spike conformations present a site of vulnerability, or a mechanism of vulnerability, I should say, to neutralization, where then we, by enforcing an RBD all-up conformation, we end up inducing a conformational state in the spike, which is no longer fusogenic. So I suggest we should look into this area specifically in coronaviruses to figure out specific means to broadly cross-neutralize coronaviruses, also in preparation for the next outbreak, which coronaviruses have been emerging every decade or so. So looking into conserved sites, I think, is very important to prepare for future pandemics. Then, as an extremely quick aside, we do also have another project by Lina where we engineered nanobody-based inhibitors, and I just wanted to highlight this preprint that is out and will soon be resubmitted after a revision. We were able to obtain significantly improved neutralization potencies by making nanobodies trimodular and also by varying the modules. So it looks like by putting 3 different modules that target the RBDs, your neutralization potency can be significantly increased over just using 3 modules that bind the same site. So unless you have a specific reason to target the site, as with the servo body where we want to be in the conserved region, you might consider using heterogeneous modules for further enhanced neutralization potency. Further, if you have small protein binders, these can be fused to signal molecules for a single-step detection system. So we wanted to present a concept where nanobodies are carrying split fragments of luciferase nanolook, and then upon binding, these split fragments come together and result in a signal. So this is a protein fragment complementation assay technique, but if you use it together with some sort of targeting module for a virus, it can be used to produce a diagnostic or a detection assay. So without further ado, I wanted to acknowledge people involved in this work, particularly Lina, my PhD student, who has been doing pretty much everything that I showed you for structural characterization, and my other group members, Ashwini and Rupesh. Rupesh is now, he was with us as a technician, and he's now taking up a PhD position in another lab, but he was also working with SARS-CoV-S trimer preparations and doing an excellent job there. And our collaborators, Kalle Saksala and Anna Mäkelä, who produced the inhibitor, and Juha Huiskonen here at the University of Helsinki, who was involved in data processing, also all the other authors of our work, and then really a very big thank to the infrastructures, which were earlier acknowledged in more detail, but here I just wanted to mention the cryo-EM unit, Instruct-ERIC, and SciLife Lab. And our funders, of course, also extremely crucial, so Academy of Finland, now Research Council of Finland, and University of Helsinki. And thank to all of you for your attention. I'll be happy to take any questions.
Okay, thank you, Ilona. So now it's a few minutes time for questions, if there are any. So at least I don't see any questions in the chat yet. So please, if you have questions, you can put it to the chat, and you can do it also during the next presentation as well, so we can then check in the end if you have any. So maybe I can make a few questions. So this multimodular system is interesting because it reminds something like zinc finger domains binding to a double-stranded DNA. And so how would the affinity change if you use such a system, given the higher degrees of freedom of this kind of linker connected domains as such?
Mm-hmm. Yes, so affinity of the individual modules remains the same, but I think where the major benefit is coming from is the avidity effect. So if we think about these interfaces, they tend to be, because they are small interfaces, so there is a high off rate in the native context. But when you put 3 modules in and you imagine these 3 modules being bound, one comes loose to the normal off rate, then it still remains in close contact very, very nearby to its binding site, and then it rebinds pretty much immediately. So I think the benefit comes from there, that once a module comes off, it is going to be rebound pretty much immediately because it's kept in close vicinity by the other bound modules. And this is a system that I think has not been taken that much advantage of earlier. Corona pandemic gave us reason now to really start looking into engineering of small protein inhibitors to target oligomeric targets such as spiral spike proteins. These are in many viruses typically different sorts of oligomers. So I think they are the prime candidate really to make this sort of inhibitor where you get the full benefit from avidity.
And how would you produce such systems in practice?
So we were just using regular GGGS linkers. So you just put typically 4 glycines, 1 serine, and you do as many repeats as needed, considering how far your targeted domains are. So the RBDs are quite close. So there you can use 15 or 20 even just residues, and that will be long enough to span the distance and allow all 3 modules to bind. If your targets are further apart, then you will need to use longer linkers. It would also be possible, I think, to use some native sequences from human proteins. For example, the GGGS linkers are not immunogenic, but if one wanted to add a longer linker, then potentially consider using a native sequence just to avoid any adverse immune reactions.
Okay, thank you. So I see no further questions, so maybe we move on to the next speaker, and then if you have questions, you can still post it to the chat window. So okay, so next one and the last today is Giovanna Viola from University of Verona, where she works as a PhD student in a group of Michael Alfag. And today she will show how to use ion mobility mass spectrometry to investigate effects of ubiquitination of conformational dynamics of amyloidogenic tau protein, which is involved in pathogenesis of Alzheimer's disease, I guess by protein misfolding. And also in this work, some Instruct-ERIC facilities were used. So floor is yours.
Thank you, Jan. So I share my screen. Okay. Do you see the presentation? Yes. Okay. Okay, so thank you. And first of all, I would like to thank the committee that gives me the possibility today to take this oral presentation. And so, as Jan said, we are going to talk about the investigation of the influence of ubiquitination on amyloidogenic tau protein by mass spectrometry. So tau protein is an Alzheimer's disease related protein, and from diagnostic point of view, Alzheimer is defined by the combination of pathological lesions in the brain made up by 2 different proteins. So amyloid plaques composed by A beta protein and neurofibrillary tangles of tau protein. Today we are going to focus just on tau protein. From behavioral point of view, Alzheimer's disease is characterized by the loss of memories, as we know, language impairment, difficulties with executive faculties, and moreover. So let's just introduce you who is tau protein. Tau is a microtubule associated protein, cytosolic protein, and what is interesting of this protein is that is an actively unfolded protein, which means that it lacks of a secondary structure. From structural point of view, is characterized by 4 regions. As you can see in the picture, the N-terminal domain, the protein-rich domain, the 4-repeat domain or microtubule binding domain, and the C-terminal domain. Microtubule binding domain because tau protein as a major function promote the tubulin assembly, so stabilize the microtubule networks, and in general it supports the neural cell function. But as I told you before, it is also involved in neurodegeneration. So when undergo to pathological condition, tau is able to form these periodical filaments. They are beta sheet structure filaments, and finally they aggregate in neurofibrillary tangles that are the toxic species. These neurofibrillary tangles are insoluble in the neuronal cells and lead the cells to the death. Okay, from now I would like to let you know how to follow tau aggregation pathway in vitro. In particular, it is very common to use this molecule called the thioflavin T because upon binding to beta sheet structure, that is our case, it displays a fluorescence. In particular, for amyloidogenic proteins, so for us is tau, the aggregation pathway is basically a sigmoidal curve, as you can see in the slide, and is characterized by 3 phases. the lag phase, in which tau is basically in monomeric state, and is also the rate determining step of the aggregation. Then the elongation phase, in which tau start to form oligomers, as you can see better in this picture, and then the plateau phase, in which mature fibers are formed. From recent cryo-EM images, we know that these beta sheet structures are like shown in the picture in the bottom of the slide. So basically, is the 4-repeat domain of tau that creates the core of these filaments by aligning this beta sheet structure, and then we have the tail, let's say, of the protein like a fusy coat structure. So since the core of this toxic species are made by the 4-repeat domain, from now we are going to be focused just on the 4-repeat domain shorter construct, also called tau K18. And in particular, in this study, we are interested to study the conformational landscape that precede tau aggregation, and even in the early stage aggregation, by mass spectroscopy. So, as an intrinsically disordered protein, tau undergoes several post-translational modifications, and here I show you let's say, the most important one, that are phosphorylation, ubiquitination, acetylation, and methylation. And you see in the picture each site that is involved in the post-translational modification. But it was found that even aberrant post-translational modification are related with abnormal aggregation of tau. And as you can see in the second picture, in particular, hyperphosphorylated tau protein was found in the tau aggregates. But besides being hyperphosphorylated, tau is also ubiquitinated in the PHF formation. And in this study, we focused on ubiquitination of tau protein to understand, as I told you before, the conformation landscape of aggregation. And in particular, what we ask to ourselves is that what are the effects of ubiquitination on tau structure, and which is the aggregation propensity when tau is ubiquitinated. So before going deep to in the detail with the mass spectroscopy data, I would like to let you know how to get ubiquitinated tau proteoform. In particular, I think the most famous and even in vitro and in vivo strategy to get ubiquitinated tau is the enzymatic reaction. In particular, for tau protein, this enzymatic reaction is carried out by a synergic action of 3 enzymes. As you can see, they are A1, A2, and A3. Basically, what happens is that the C-terminal domain of ubiquitin is attached to the lysine group of the substrate. In our case, is tau. So, in normal condition, this ubiquitinated substrate goes to the proteasome, the ubiquitin chain is detached, and the protein is degraded. In our case, we want to observe the pathological condition of ubiquitinated tau proteoform. But the problem of enzymatic reaction is that if you think about just tau 4-repeat domain, it has 20 lysine residues, which means that if we use the enzymatic reaction, we get a pool of mono-ubiquitinated substrates. But in this way, we lost the site specificity of ubiquitination impact. So that's why we moved to the semi-synthetic ubiquitination, and in particular we started from a ubiquitin-intane substrate. We get this ubiquitin modified protein with an amino tantiol group as a flag, and then we activate this thiol group with an Hellman's reagents, and we get this modified ubiquitin. Sorry. After that, we choose a precise mutant of tau on which we put a cysteine on the amino acid on which we want to attach the ubiquitin, and we let react the ubiquitin with the tau mutants, and in this way we get a monoubiquitinated tau at the desired position. So in our group, actually, we already performed some studies about monoubiquitinated and deubiquitinated tau in position 353 with different techniques. Here I show you the THTSA and some transmission electron microscopy images. Briefly, what we observed is that ubiquitin at position 254 and 353 is able to slow down the aggregation, but not arrest defibrillation. Instead, ubiquitination at position 311 is able to inhibit the formation of fibrils. Both this information coming from both THTSA and TEM analysis. Okay, so starting from these results, of course, we wanted to shed light on what happened at molecular level when tau is ubiquitinated and when we let him aggregate. And for this aim, we decided to use mass spectroscopy. In particular, we combine two let's say technique. Okay, so the first is the first idea was to acquire native mass native mass spectra on different tau ubiquitinated tau proteoform. Why? Because in native mass spectroscopy, basically what is normal to do is that so we use a volatile solvent, because in this way we are able to retain the native structure from solution to gas phase. And here I reported you an example of two different proteins folding. So the unfolded protein shows this profile that is characterized by an high charged peaks with several peaks and the low m over zeta values. On the other hand, the folded protein in native condition displays this narrow spread distribution with low charge states and high m over zeta value. With the native mass spectroscopy, we wanted to combine also ion mobility data. Why? Because, so basically how ion mobility works. The sample is injected in the analyzer tunnel, and in here what we have is the electric field that push the ions through the detector, but we have also a gas flow that goes in opposite direction with the electric field. So what happens is that, let's say, open-head structure feel more resistant against the gas flow compared to compact structure. So basically what happened is that compact protein, in our case, reach faster the detector compared to the open-head one. So to let you better understand how is powerful this technique, I show you here an example. So as you can see, we have three molecules with the same molecular weight, because these three molecules share the same elemental formula. And so from a typical, let's say, classical mass spectrum, we cannot separate the three molecules since they have the three masses. So mass spectra are mass dependent. But with ion mobility, since they have different spatial conformation, we are perfectly able to separate the three molecules, as you can see in the picture. So our idea is to combine the information from native spectra with this collision cross-section value that are extracted from IO mobility, because collision cross-section is mass and shape dependent. And what is very important is that these collision cross-section values are quantitative values. So this means that they can be compared after with theoretical values from DFT calculation, for example. Okay, so briefly, I will show you the really first preliminary data we acquired at University of Eastern Finland under the supervision of Professor Yannis. And here I show you two native mass spectra. So on the right, you can see the native mass spectra of ubiquitin. Ubiquitin is the prototypal folded protein. And in fact, as you can see, we can observe a few peaks with low charge states and with high m over zeta values. On the other hand, on the left of the slide, you can see the native mass spectrum of tau protein. And here the things are a bit more complicated, because even if we can observe the typical spread profile of an unfolded protein, we can also see other distribution. So is in some way this spectrum reveal that in some way tau protein has like an extended population and a partially compact structure. So it's an ensemble actually of population. Then we acquired also native mass spectra on mono and demonoubiquitinated tau. Even here you can see there is an ensemble of structures. And finally, we acquired the first data about IO mobility. Again, on the you can see the native mass spectra of tau protein, and we extracted the CCS values for the more charged states of each of the distribution, let's say. As you can see, in fact, we obtained we got 3 different values of CCS. So since this number is related to the shape, okay, to the structure, spatial distribution of the protein, this means that actually we have 3 different populations of structure for tau protein in monomeric state. Then we acquired another, we acquired the native mass spectrum of monoubiquitinated tau and IO mobility values. Even here, as you can see, we got 2 different values for the 2, let's say, distribution you can see in the spectrum. Okay, so in conclusion, I can say that for tau aggregation, so we observed the tau aggregation and in particular post-translational modification, because they are crucial for neurodegeneration. And in this work, we focused on ubiquitination of tau. We acquired some native mass spectra, since native mass mass spectroscopy is able to preserve the native states of compounds, and in our case of proteins. And we decided to combine this information with IO mobility, because IO mobility is able to provide 2D separation by M over zeta and CCS values. Right now, we got really interesting preliminary data on monomeric states of tau protein and its ubiquitinated mutants. And for sure, for the future, we would like to acquire IO mobility, so mobility information about the early aggregation states of tau. So just to understand about the oligomeric states in the beginning of the aggregation curve, and absolutely we will need to confirm to compare these CCS values we got by mass spectroscopy with DFT models, so by theoretical with theoretical values. Okay, I would like to thank my group in Verona and Professor Jan and Dr. Mikko that supports me with my period abroad in Finland, and you, of course, for your kind attention.
All right, thank you, Giovanna.
Okay.
So, of course, I'm more familiar with this work since the visit was to our lab last summer.
Yeah.
So, but anyway, if you have some questions to Giovanna, please use the chat to put your questions there. So maybe I have a more like so I can start with some more like a personal question. So. So once the first visit was over last summer, so how do you feel this training provided by the access visits, since you're doing PhD now, was helping improving your skills in your research?
Yeah, for sure, it helps me a lot. Yeah. Sorry, I can. Yeah. Just a moment. Okay. So yeah, it was a great period because I discovered this new technique, the IO mobility, and I guess for our aim is really a powerful technique because okay, now they are just preliminary data, but after that, when we will start to observe aggregates, or at least early stage, I guess we will get really interesting information.
Yeah. So yeah, I agree. So one of the strong points in having these kinds of access visits is indeed to learn some new techniques which may not be available in your own laboratory or even your own country. So and this is all provided by Instruct, and also some funds are given for these visits as well. So okay, so there are a couple of questions in the chat, so maybe I can read these for you. So a couple of questions to Ilona first. So there is question: did you tune the linker lengths, or would you do that with the knowledge of the structure?
Right, so here the inhibitor was generated with 15 residue linkers, and looking at the structure so that was already at the point when we did not have structural insight yet as to where it is binding. I think we just kind of lucked out in this design because 15 residues is extremely suitable considering that the RBD distances from each other and the particularly in the up state. One could probably use a little bit shorter as well, considering that the up state RBDs do present the sites quite close to each other. So that is a possibility. But I would say in the case of SARS-CoV-2, it is maybe less crucial because the sites are going to be close. But we do have other projects where it is quite important that we carefully measure the distance and then tailor the linkers to match that.
Okay, thank you. Then there's another question from the audience. Are the Sherpa bodies only suitable for respiratory viruses, or can these be designed to target intracellular viral components such as RDRP?
So they can be designed for anything, essentially. We do have that discovery library, so if there is a recombinant protein, then Sherpa bodies can be pulled down. It seems to be a very prolific system for generating these sort of protein-protein binders, most likely because SH3 domains, even in their biological native function, they are mediating protein-protein interactions with these regions that now have been diversified in this library. Maybe one thing to keep in mind, though, is that we wanted to target the spike to make the virus incompetent of initiating infection. If we were targeting an intraviral component, then it is unlikely that we would be able to get the inhibitor inside the viruses very easily. So then instead of, because the spike is at the surface, so if you, let's say we did do this in a mouse model, you give mice a virus challenge, and then you give them the inhibitor, the pathways, you know, the virus is going to be present there, the spike is going to be exposed, you can effectively inhibit the spike. If it is an intraviral component, then it becomes more of a question of getting the inhibitor into the cells, which can be done as well, but it's not something we did in here because our idea was to prevent the virus at the earliest stage, almost as a prophylactic, that you could potentially take the servo body via an inhalator and then be protected from catching the virus in the first place, because you have the inhibitor present in your respiratory system, and any virus that comes in is going to get neutralized before it can do anything. If we targeted something that already then infection has to be going for us to target it, then we are targeting at a little bit later stage. So yeah, I think for viruses, we'll want to neutralize them at an early stage. For any sort of other disease, though, we might consider trying to inhibit cellular processes that are etiologic to a disease.
Okay, thank you. Then there's one question to Giovanna. So
Yeah.
can you measure MS with time, EI at different times of aggregation?
Okay, so thank you for the question. Actually, at the end of my period abroad, we started to, let's say, to observe the aggregation along the time. But the problem right now is that the aggregation buffer is different from the ones we use for the native mass. So for sure, we need to adjust the method, but actually is what we want to do. So to observe the aggregation along the time, yeah, we need to improve the method, but for sure is the future.
Yeah. So basically, this is doable. Well, it also depends on the time frame that it takes to something to aggregate. And so if it's slow enough, it can be done in a conventional fashion. And if it is faster, then there are some other options available, like stop flow connected to the mass spectrometer, and then of course something can be maybe done with the solvent being the other hand suitable for these aggregation studies, as well as in the end suitable for the mass spectrometric analysis. So there are some ways, but let's see. Time shows. Time will show.
Absolutely.
All right. So I don't see any other questions. John, do you see any other questions in the chat, or?
No, I think we're clear.
Okay, so if no further questions are from the audience, I guess it's time to thank both speakers of today, Ilona and Giovanna. Thank you very much for accepting the invitation to speak here today, and we'll thank all the participants for today. So thank you, and remember to apply if you are interested about Instruct-ERIC services. So you can approach any of us for any further comments, of course. And have a nice day, and see you in next webinar. Okay, thank you.
Bye.
Bye.